welcome everybody to the final Evenings at Whitney of the spring series. And today I'm delighted to be introducing Dr. Max Stammnitz to you. So I've known Max for about 6 years now, and I've followed his work closely. And I think after the talk, you'll agree it's fascinating work that he does. And he's been collaborating with me, but also a number of other staff members here at the Whitney lab for those 6 years in a whole variety of projects. So obviously his work overlaps a little bit with what we do here at the Sea Turtle Hospital. And I'm also delighted that some of the past employees who worked with Max have been able to make it here tonight to see his talk as well. Max received his undergraduate degree in his native Germany from the University of Freiburg. After his undergraduate, he moved to the UK, to the University of Cambridge, where he conducted his PhD in the computational and maths department there. But actually, although he's mostly a computational biologist, he does a lot of field biology and lab work as well. So while he was in Cambridge, he was also working very closely with their vet department and also the world-renowned Sanger Sequencing Institute. So he's really an expert in everything from field work all the way through to high-end computing as well. And then after he completed his PhD, he received a prestigious European Molecular Biology Organization fellowship, which enabled him to move to Spain, where he currently resides, where he is working at the Genomic, the Center for Genomic Regulation in Barcelona. And although he's an early career scientist, he consistently publishes work in really top tier journals. In fact, the only other two people I know who have as good a publication record as Max are our very own Dr. Martindale and Dr. Morose. So I think he's in good company. And I think he's destined to continue to do great things in the very near future. So his latest high impact paper came out only a couple of weeks ago, so it's super fresh, hot off the presses, and we're lucky that he's going to be able to tell us all about it tonight. So get ready to hear everything that's worth knowing about the debilitating Tasmanian Devil Facial Tumour Disease. So welcome, Max.
Thanks, Dave. Well, can you hear me well? Yeah. Thanks a lot, Dave, for the very flattering introduction. It's a great honor to speak in front of you tonight. It is really my very first talk in the United States, my very first time in Florida as a visitor. So I only had this morning and afternoon to look around, and I have to really admit this is a wonderful place, both to live and do research. I can tell at first sight, you know, like getting up in the morning and visiting. Yeah, well, so lovely place to be. What I'm going to show you tonight is not always like seen as something beautiful. We're talking about a very unusual pathogen, transmissible cancer in Tasmanian devils. It is not very beautiful from a like visual perspective, but through an evolutionary lens, I hope to convince you that it's a story really, really worth having a look at. Genetics wise is a truly fascinating entity of disease. And it is structured in the sense that I will want to walk you through the natural history of Tasmanian devils. I'm not sure how many of you have been to Tasmania. I guess very few, because it's a very remote island, pretty much on the other side of the world from here. And once we go through the Tasmanian devil story, then I intend to walk you through, well, contemporary thoughts of cancer evolution, both in humans and then applied to Tasmanian devils' rare transmissible cancer diseases. So this is Australia. And down here, separated by the very shallow Bass Strait, is the southernmost Australian state of Tasmania. It's a relatively small island. That separation only took place around 10 to 12,000 years ago, after the ice ages when the sea levels rose. So many of the animals down here, they basically passed through a population bottleneck, and Tasmanian devils are one of these species. So they used to be present all along the southern hemisphere of Australia, but as it got hotter over the last few thousand years, and through the introduction of dingoes on the mainland of Australia, but not in Tasmania, we believe that these are the main reasons why they became extinct on the mainland a few thousand years ago. whereas in Tasmania, they very much continue to be alive. And how do they look like? Not like this. I guess that might be a character you've seen in movies or your kids' movies. They're in fact not brown and on two legs, but they're rather black, walk on four. They're about this big, and adult males can weigh around 13 kilos, so relatively heavy, like small-sized dogs, with a distinctive feature of a very large head. And what do they need that head for? They're marsupial carnivores, so the largest marsupial carnivores on Earth. So they're scavengers. They fight over carcasses in the middle of the night, and sort of an ecological cleanup service to the environmental, the ecosystem of Tasmania. They're marsupials, so when they're very young, so about this tiny, like they fit on a little coin, they are born, and then they crawl up this pouch where they're weaned by their moms. And once they get a bit older, they start showing these behaviors. So Tasmanian devils, I guess that's where their name originally comes from. They're known to be vicious amongst each other when it comes to fighting for food and mating partners. I tend to show you a little video of two Tasmanian devils interacting. Hope that works out sound-wise. Like, so Tasmanian devil fight. There you go. Okay, so I was told these two are actually friends from captive facility. But yeah, these are the sort of interactions that we do observe in nature. Towards humans, I have to say they're incredibly tame. You can handle them, like vets are regularly trained to look after them in the field. And I had the honor to go to Tasmania twice and do a bit of field work with our colleagues. What we tend to do is we set up these large plastic pipes, relatively solid plastic, and then put a meat carcass in there. And then at night, because these are nocturnal animals, Tasmanian devils, they have really good smelling instincts, and they walk along the forest corridors of Tasmania, smell the carcasses, go into these traps, and once they pull the string, the door closes. And then the next morning, we tend to come along. We put them in a little bag, and then from the bag, you sort of examine their facial region to look for Devil Facial Tumour Disease, the transmissible cancers that we're going to talk about in a minute. Right, so much for the Tasmanian devil. This species has really severely declined in the last 20 to 30 years. With the onset of these transmissible cancer diseases, we have seen that populations all over the island of Tasmania have actually crashed to the level of where it's now listed as an endangered species on the Red List of the IUCN. But before we get to their disease, I thought I'd walk you through a bit of cancer evolution theory quickly. So what is cancer? Just very basically. There's lots of different descriptions of this. Some people would say cancers are sort of parasites growing within. So cells in our body, in animals' bodies, that take over the body itself. So when part of the body becomes autonomous and uses more resources than it ought to use, that's the basic idea of what a cancer is and what a tumor is once it evolves. And through a molecular lens, how we think it happens is usually that we grow from the embryo stage. Our cells, they acquire mutations throughout our lifetime, and then some of these mutations, indicated here by a star, they may give cells in our bodies a growth advantage over their environment, over other cells. So certain types of mutations allow cells to proliferate more than they ought to do. And that goes on. Once you have a faster division rate, you're more likely to accumulate additional mutations, and so on, to the point where you start growing a tissue within a tissue. And that then later can turn into, well, initially benign and then invasive tumors. So there's a model of cancer evolution pretty much drawn in the 1970s, which still holds to the current day as we understand it. From a normal cell in our body, cancer takes a branched evolutionary process with several dead ends. So once you accumulate mutations in your cells, some of these mutations might be lethal to the cells. But along the evolutionary path, there could also be several advantageous mutations that then confer a strongly selective growth advantage to the point where you outgrow the tissue. But the key point is that as far as humans are concerned, tumors die with the patients. So the dead end of evolution, of this selfish evolution within our bodies, is the dead end of our own body. And in humans, as I said, we don't have an idea of cells jumping from one individual to the other. Whereas in some animal systems, we do in fact observe this cancer evolution to continue beyond the original confinements of the body of the individual that obtained the cancer in the first place. So essentially at the outgrown tissue stage, what we observe is that cells are able to jump, to transmit from one individual to the next, like an organ transplant. So these are cancer organ transplants, like a very horrible idea. Luckily for us, we've only so far observed this in the animal kingdom in relatively few species. 2 of them are mammals. So the canine transmissible venereal tumor, it's a genitally transmitted, sex-transmitted disease that affects stray dogs populations all over the planet. Wherever you have like a large amount of free-ranging dogs, this disease is usually present. Places like India, Colombia. So we could reconstruct that history of spread across the world with people actually migrating to different places and bringing their dogs with them. Then another disease here is bivalve transmissible neoplasia. So this is a more recent observation. We know that across the world oceans, there are several species of shellfish or clams, cockles, oysters that can shed transmissible cancer cells out in the seawater, and then these can be absorbed by other individuals, and these cells continue proliferating. That's really puzzling. And then the third instance is the one that we're going to talk about tonight, Tasmanian devil transmissible facial tumor disease, which manifests as these facial tumors. So this is a really disfiguring image. I don't intend to share worse ones with you tonight. If you want to look at, how the disease can manifest across individuals, just Google it. But essentially, these tumors are characteristic by growing in the facial area, also inside the mouth, and slowly taking over the host's body. So usually within a few months, that leads to the death of the individual. It spreads. So what was first observed in 1996 up here in the northeastern corner of Tasmania by a wildlife photographer was initially thought to be just a wildlife cancer event. So that person saw some individuals caught tumors in their faces and thought it was pretty much a lone instance. But then in relatively little time, over the course of just around 25 years, we saw these types of facial tumors appearing in many Tasmanian devils all over the island. And what was then really puzzling is a discovery that the chromosomes of these tumors, so when you take biopsies, you put them in a culture dish, you extract the chromosomes and do flow sorting. What you see is that all of these tumors, no matter if you separate it from the south or the north or the east or west, we don't have tumors so far, but from here, then these have the same set of marker chromosomes. And that indicated to us that it is in fact the cells themselves that are the infectious agent. It is not a virus that spreads between Tasmanian devils that causes these diseases to flick up, but it's the cells themselves that are the vector. And as if that wasn't enough, in 2014, in the southeastern part of Tasmania, people observed a second instance of transmissible cancer. So how did they realize that? So essentially they saw animals having these facial tumors. And at first they thought, okay, that's yet another case. in this well, huge epidemic. But then they realized when looking at the karyotypes that these tumors down here in the southeast, they also carried a Y chromosome. So unlike the first transmissible cancer that had been seen, where we haven't seen a Y chromosome, and where we are pretty much sure that it originated in a female individual, this one here very much evolved from an ancestral male host with a Y chromosome. So there's a few really puzzling questions here. Like, I mean, the transmissible cancer is rare enough, and well, we've barely observed this in nature before. So why would it happen twice to the same species? What is their vulnerability? What is the problem with the Tasmanian devil? Could it be that humans have like through some environmental contamination or through the introduction of a parasitic species or another oncogenic trigger have caused this? Or is it like a genetic vulnerability in this population, maybe from the founder event that led them to be cut off from mainland Australia 2,000 years ago? We don't know. So we really set out over the last 5-6 years to do a genetic characterization of these tumors in depth, and not just using the traditional methods of karyotyping, as I'm showing here, but actually looking into the exact positions on every single chromosome, and then reconstructing the evolutionary history from these tumors. So the idea is that you trace back the mutations of these tumors as you go on with time. You trace back to the very point where these cancers arose, and you try to find out what impact mutations have had along their lifestyle and lifetime. But this is very much also a non-model species. So, Tasmanian devils are not animals that people have in the lab. So in terms of the genetics, we first had to basically assure that we have the right resources to do these analyses. And back in 2012, my PhD supervisor, she published a paper with the first Tasmanian devil reference genome. Back then, done with short read sequencing, Illumina sequencing, reads only about 100 base pairs of DNA stretches long, covering these 6 major chromosomes and chromosome X. And then she generated a first, what we call reference genome of a normal healthy Tasmanian devil. But it consisted of roughly like 35,000 little pieces of genetic information all over these chromosomes. And when we set out to do this characterization between tumors, our concern was that important information might be lost in these gaps. So one of my first projects was to actually to generate new genetic data to close these gaps. And what we've done for that is mainly nanopore sequencing. So Dave here in the audience is using nanopore sequencing a lot nowadays. But back in 2018, this was still a really young technology. And the first protocols for doing these extremely long DNA sequencing strands, they had just come out. So with this technology, we can go way beyond the 100 base pair threshold. We can sequence DNA molecules of up to 1 million base pairs in length. So that's really powerful to close and improve on our reference atlases. So to make a long story short, I mean, this took us several years with help from world experts in DNA reference genome assembly. But we managed to close almost all of these gaps in the Tasmanian devil reference. And with that, we then set out to do our actual comparison and trying to understand what's going on in these tumors. So this is our study cohort. With the help of truly amazing fieldwork collaborators over Tasmania, we could collect a data set with 78 DFT1 tumors from the first transmissible cancer that was first observed in '96, and that's now spread all over the island, and around 41 tumors of the second instance of the disease that was derived from a male individual. And what we then did was extracting DNA from all of these biopsies and put them on a Illumina sequencing instrument. So this is, again, short read sequencer. We sequenced these tumors to around 83-fold depth. That means every single genetic position that we can track has on average 80 times sequencing coverage, which is really important to get accurate estimates of mutation burden across the genomes. And we also sequenced the associated hosts to basically make a comparison of how these tumors sort of like cluster away from the normal Tasmanian devil population. And we gather all the mutations from all of these tumors. Again, I'm leaving out technical details here, but you can ask me later if you like. So we are basically extracting all the mutations that are unique to just the tumors, but that are absent in normal individuals. So it's not a population study, it's a tumor phylogeny that we're seeing here. So with all of these DFT1 tumors collected all across the island and with their dating information, as in when we collected them, we could reconstruct their phylogenetic tree. So that led us to a rather unexpected time of origin estimate around the 1980s. So as I said, it was first observed in 1996. So we were quite surprised that it seems there was a stretch of around 10 years in which this disease must have been present at a low level in the population, but wasn't observed. And then again, we saw that at the very onset of the disease, there was this kind of explosive event where what we now see in terms of the diversity of this disease, there has been like a spreading where a single individual probably bit 5 to 6 other Tasmanian devils, and each of these carried the disease into a different direction of the sky here in the northeast. So one lineage, for example, is clade C down here, started migrating westwards, whereas the other 3 to 4 remained in this area. That story is a bit different for the second instance of transmissible cancer. So we see that this only like splits into 2 major clades, which we term A and B here. Yeah, so what can we do with these trees now? We want to compare these 2 diseases. So one obvious thing to look at is the speed of evolution. So what we do here is a correlation of different types of mutations that accumulate over time. So substitutions are essentially point mutations, so just single base substitutions in the genome, and indels are short insertions and deletions. And as you can see here is basically DFT2, the second instance, appears to mutate considerably faster than DFT1. So it has higher evolutionary speed. But correlations are not always that good. So if you look up here, there's quite a lot of variation. And we looked into this in a bit more detail. So basically, we were trying to explain how could it be that the mutation rate is somewhat linear, but starts fluctuating at times. And if you look into the tree, if you have that information of phylogenetic composure of evolution of these cancers, then you can see that in certain branches, it seems that the mutation rate has actually slowed down. So with this westward migrating clade of DFT1, we see that on average, they lack around 1,000 variants. And that means that very much in this branch where DFT1 migrated westward, we think that, well, there could be several explanations for why that happened, but we think that this leads to sort of transient fluctuations. So the longer we sample in time, the more ambiguous these measurements become. But so far, we think our estimates are pretty good, and they allow us to reliably trace back the origin to the 80s. But there's one important exception, which I didn't put on this plot. There's one tumor, which is pretty much from here, whose mutation rate is somewhere up north here. So it has way more mutations than all of the others. So we call that the hypermutator DFT1. It had a very different mutation profile, very dense mutation load all over the chromosomes. And we realized that most likely this is due to a deletion in a gene called MLH1. So this is a DNA mismatch repair protein, which has been shown to be absent in human cancers that also have this elevated mutation burden. So this is a unique deletion only present in this sublineage. And you can speculate as to why we've only been able to sample one of these tumors. Maybe it's become extinct, not necessarily just because of its high mutation burden, but there seems to be something really odd about this ancestral clade. Then there's another type of mutations that I want to walk you through, line 1 transposons. So line 1, these are transposable elements that also humans have and many other species. So these are roughly 6 kilobase long loci in our genomes that can self-transcribe. They basically code for 2 proteins that then bind to their own transcript. And with the help of these proteins, they can migrate back into the nucleus and kind of hack their way back into the genome. So it's a retrotransposon. And this retrotransposon has been shown to be active in several human cancers over the last few years. So we were curious to see if with the help of this new reference genome, we could also detect this activity in our transmissible cancers. And indeed we did, but only one of them. I can't tell you why that is, why these transposons are so active. And that's an ongoing investigation. But what I can tell you is that there's one locus. So at the end of this chromosome 1, so this is basically all of the Tasmanian devil chromosomes represented by a circular map. And this is the source locus where the original transposon was seated. And then it seems that this is a locus that's highly transcribing, firing out copies of itself, which then insert at random positions all over the chromosomes. And you can actually trace this specific source locus from the origin of the disease all the way through the phylogeny. It's a really puzzling result. And there might be several reasons for that. Maybe the promoter of this is highly active because it's not methylated, as transposons usually are methylated to be silenced. So, but maybe the cells are also prone to just by their original state to acquire transposons because of their chromatin or other cellular factors. There's more big mutations. At the very beginning, when DFT1 and DFT2 arose, we see highly dense mutation loads. So that's again these circular representations of the chromosomes. And with these arcs, we represent exchanges of genetic sequence within and between chromosomes that are indicative of certain processes that went on. So we very much think that at the beginning of DFT1, there was a sort of catastrophe going on at the telomere of one of the chromosome 1s. And that maybe in the eye of the storm here, there sits an important gene that could explain why the disease went forward and the cell simply didn't die. So and the other instance, DFT2 also has several of these translocations, but not as clusters as DFT1. And as you go through the tree, you can see these types of events happening at different sections. I mean, you see these clustered events, like weirdly like interacting chromosome pieces that exchange information. So this is far away from normal cellular development. This should be checked. The DNA repair should account for this, but these cancers continue to proliferate and acquire these large mutations. Tendentially, we think still at the chromosome ends. Like this is a good example where almost all the chromosome ends are involved. And sometimes they form these circular loops. So we think multiple chromosomes might be involved at once. So these are phenomena we've also seen in human cancer. So these are not new observations, but our model system allows us to follow these processes over time. And see how that might alter the cellular path along these along their fitness trajectories. Another large-scale event, which is called whole genome doubling. That's something we see mostly in these DFT1s. So this is when basically the cells, as they divide, they not only divide and form 2 daughter cells, but they double the content of their genomes. So rather than having 2 original copies, they have 4. There's several theories as to how this can accommodate cancer evolution in the long run. So this might buffer for deleterious mutations. But again, we don't fully know why this continues to happen and how, and we don't know the exact impact, but we can just observe it and follow it. And in some instances, we've seen that happened several years ago. So this, is again, pretty much like was not expected from our end. So what about the genetic impact? So I've talked about types of mutations, about like DNA alterations on big scale on the chromosomes or small point mutations, indels. But what people usually want to know is what does it matter to the cell? So what are the genes affected? How could these events help the cells divide faster or overcome transmission barriers? So how are these cells able to overcome the immune systems of the hosts and so on? So what we did was what is called a DNDS analysis. So evolutionary biologists in the audience might be familiar with that. Basically, we look at mutations. So in this case, point mutations and indels that happen only in genes. And then we derive a ratio of mutations that are non-synonymous. So they cause an exchange of an amino acid in the protein coding sequence with mutations that don't do that, which we call synonymous ones. And usually, well, the background model is that if that ratio equals to 1, we see more or less the same number of mutations that are synonymous and non-synonymous, accounting for a few like covariates in the genome. That suggests that these cells are more or less neutrally evolving. There's no strong selection pressure on them. And we see that for both of these transmissible cancers, it doesn't look like there is an excess of non-synonymous mutations. But a few very specific genes have an excess of mutations. For example, in DFT1, we see that this MGA has a mutation at several positions in the tree. So given that there's just around 2,000 mutations encoding genes all over this tree, and the Tasmanian devil genome has around 19,000 different genes, it's highly unexpected that this would happen by chance. And another thing that I want to follow through is losses of chromosome Y of the originally male-derived DFT2. We also see that happening quite frequently. So what is MGA? Well, it is a gene that suppresses MYC activity. MYC is a famous protein, especially for developmental biologists and people that work with stem cells. It's one of those key transcription factors that, like a master regulator, as we call it, it can switch on cellular developmental programs and is known to be involved in several human cancers. Because once you switch on MYC, once it's heavily switched on, the cellular path can be heavily altered. And we know that MGA is a suppressor of MYC. So if you switch off the suppressor, you switch on the protein that's supposed to be suppressed. And all of these mutations are basically truncating. So here's the start site, and these black bars are the exons. And we see that there's several of these frame shifting mutations happen all over the tree, and stop sites in positions where so we basically cut the protein in little pieces, which probably renders it unfunctional. And as we were like wrapping up this story, we saw a paper coming out because MGA is not an established cancer gene in the human literature, but it is arising more and more interest in the community. And there was a mouse study where like people have followed a mouse lung cancer model. They knocked out this MGA gene in the tumors and saw that the survival time, the average survival time decreased. So it really seems that this is a gene that increases proliferation in certain types of tumors. And it's also observed to be deleted in around 10% of human lung cancers. So not enough to be put in a gene panel, but a candidate of high suspicion in the community. And we think that our transmissible cancer trees actually strengthen the argument for this gene to be put into the screening panels. Then the other example I want to talk to you about is this frequent chromosome Y loss in DFT2. If you look at the samples that we've collected over this 5-year time span, you see that at first, as you go from through the timeline here, at first, most of the tumors or all of them carried a Y chromosome. That's why we found out that it was from male-derived cell in the first place. But as we keep on sampling, it seems to disappear. Most likely because in the ancestor of this clade B, it's been lost. And this looks a bit like a selective sweep to us. So maybe the Y chromosome is actually a hindrance to these cells. And why would that be? Well, what we've seen is that unlike DFT1, DFT2 heavily infects males. There's an excess of male hosts over female hosts. And we believe that that has to do with the antigen machinery of females. So a Y chromosome is, I mean, it's not a big chromosome, but it encodes for proteins that should be absent in females. So if a female individual receives this tumor, this tumor allograft from a male cell, then the Y chromosomes might shed antigens into the body that are recognized by the immune cells. So that might be one of the few markers that these cells actually have on their surface and whereby they might be recognized. So from an evolutionary parasitic point of view, it's beneficial to the cells probably to get rid of that Y chromosome and thereby double its pool of potential future hosts. We cannot fully prove this hypothesis. It might also be that the Y chromosome is just more likely to be lost by chance. But I have to say, we know people that have cultured these cells in the lab over several, like dozens of passages, and they've never observed the Y chromosome loss. So I would very much hypothesize that that is in fact a functional mutation that we're seeing here, and that with time, as we continue sampling, we may very likely see more females being affected by that disease, but time will tell. And then a last story. As we went through these gene tables and trying to find out what the functional meanings of these mutations were, we realized that actually between these two tumors, there's almost nothing in common. I mean, I've shown you big scale mutations examples. I've shown you these transposon activities, but the genes that are in all of this, I mean, there's not that many mutations in genes to begin with, but there's zero overlap between the two cancers. And that was not expected by us. We thought very much, okay, we might see the same instance of a disease twice. But although they look exactly the same on the animals, it seems that they're operating through slightly different molecular wirings. But there's one pair of genes which are quite closely related in terms of their protein sequences, the so-called platelet-derived growth factor receptors B and A. So they're in different positions on the chromosomes. And what we see is that in one of the tumors, in one of the cancers, the beta form of the receptors heavily gained. So across the evolution, across the tree, we see several amplifications of that gene and very high expression. So probably it's a very active receptor, like in excess present on the cell. And similarly, we see the other receptor being heavily amplified in the other type of tumor. So when we put these cells into culture dish, I should say that these receptors can heterodimerize and or homodimerize, and once they become active, they trigger cellular proliferation. And they're mostly known to be active in wound repair processes, but also again in some human cancers. And that's why there's drugs against these. So they're on the cell surface, and we can try to inhibit the activity of these kinases. So we tested that in an unbiased way. So you can culture these types of tumors in a dish. And again, with some collaborators at the Sanger Institute, we used a robotic facility we basically grew these cells in Petri dishes, and then we titrated a panel of more than 100 chemotherapeutic agents at different concentrations into these, and derived a dose response profile for each inhibitor. And I'm not showing you the full result set here, but I want to focus on these key inhibitors that are known to be like associated in PDGFR inhibition. That's, in fact, we knew that we had that result already a few years ago, before we built these phylogenies and before we saw all of these amplifications on the chromosome. So these really two strands of evidence coming together and suggesting that these receptors have a property of driving the cellular growth machinery forward. When we came up with this, some rather sensationalist newspaper, suggested that we have found a cure now. I think we're very far away from that. There's no cure yet for this disease. Even if there were, I mean, how do you administer that to wild population of animals? Not only through a logistic lens, but also through an ethical one. I mean, should we do that? I mean, isn't it a wildlife disease? I've shown you no results of potential human implication because we didn't find any evidence in the genomes. you can look for unusual mutation types that are usually caused by exogenous agents. But all the mutations, especially the point mutations in indels that we find in humans, they're very much known to be part of the endogenous machinery. It's very normal cellular development and division that leads to these mutations. So we could not find any implications of human activity in there, which doesn't mean that there wasn't, but there's a big question of responsibility here. As conservationists, we certainly do want the species to survive in the long run, so we should continue monitoring it. There are like ongoing activities. There's people developing a vaccine, or have been trying to do that for many years. But we think at least we've understood a bit more like how these cells might actually grow and continue to divide by looking at these PDGF receptors. And maybe they hold the clue, in future to also understand more, like how we could possibly interact with this disease. Okay, as a summary, wrapping up, 2 transmissible cancers. We've tried to track their evolution by using high throughput genetics, by sequencing these tumors, generating a huge data set. We've had to use supercomputers, and as Dave was saying at the beginning, we're a very interdisciplinary team. We had computer scientists with us, mathematicians, of course, ecologists and veterinarians. So this was a very collaborational project with parties both in Australia and the UK. And together with all of these people, we've come up with these results. So we believe DFT2 has a capacity to evolve faster than DFT1, that it's an agent that once it escapes this small zone where it's currently held, this southeastern peninsula, once it escapes that, it has all the tools to become yet another global pathogen in terms of the Tasmanian devil population. We realized the disease can remain hidden for a while, despite the fact that there are ongoing fieldwork efforts. At low level, there might be other transmissible cancers that we're not observing. And some of them might die out before they become big pathogens, before they become widespread. And some of them might make it as these 2 clones that we have in front of us. We have seen a hypermutator phenotype, very unexpected, and it's presumably become extinct in DFT1. We've seen MGA mutations, so to us that's very interesting finding, because we feel like we can strengthen the claim that this is a cancer gene worth looking at also in humans. We've found line 1 transposon mutagenesis in DFT2. I didn't say that before, but it was previously claimed that these transposons altogether are absent, are not active in the Tasmanian devil as such. So not in germline, that it would be an unfunctional type of biological entity, and we can clearly say that that's not the case, that in these tumors they're highly active and probably therefore also to some degree in the ancestry of Tasmanian devils. We see these Y-chromosome losses in DFT2. I believe, and we believe, I guess, that this may indicate an adaptation of the disease to become more infectious towards females. And we've found that there's this intriguing PDGF receptor signaling in both diseases, really the only sort of cellular link that we see in terms of the mutational landscape. And that could indicate a vulnerability worth exploiting in the future. Not sure through the conventional drugs that we have, but maybe people can devise a smarter strategy. As Dave was suggesting, we've just published the summary of most of the things I've shown you in Science a few weeks ago. Please have a look at the paper if you want to learn more. Can also talk to me later if this was unclear to some degree. I hope I could keep you all on board. And that leaves me thanking, first of all, you for coming here tonight. Dave for inviting me and bringing me over. All these people who contributed to the project over the last few years. So Liz Murchison really is a founding figure in that field. She was the first person to sequence these tumors, and has really taught us all a lot about how to analyze cancer genomes, how to do devil sampling. So she's brought us over to her native Tasmania. We've had these fantastic collaborators at the Sanger Institute near Cambridge who were just, we're currently driving forward the efforts of doing reference genome resources on many, many more species beyond the Tasmanian devil. So we've had the opportunity to learn from them and to integrate these new DNA technologies that can sequence extremely long molecules. So it was like a very rich experience to do PhD, I have to say, and I'm extremely grateful. Having had this adventure, and with that, I hope could get you excited about the topic. And thank you very much.